Hello, everyone. Happy Thursday, and we welcome you all back to Science in the Age of COVID-19 seminar series. Thanks for joining. Schools, colleges, workplaces, economies, and societies have either opened up or are slowly opening up. At this point, we all agree that a rapid, reliable, high-throughput testing scheme is critical in order for us to do all this safely. We've talked about testing in the past in our talk series, and we will be revisiting the topic today. It gives us immense pleasure to welcome Dr. Sri Kosuri to our seminar series today, who's going to introduce to us a scalable, low-cost, next-generation sequencing or NGS sequencing platform-based testing strategy called Swabseq. A little about Sri before we get on. Sri is a co-founder and CEO of Octant, a small molecule drug discovery startup applying advances in synthetic biology, genomics, and computation towards the treatment of complex diseases. Sri is on leave from UCLA, where he's an associate professor in the chemistry and biochemistry department. Several feathers to his cap, Sri has helped build several technologies, labs, and companies in synthetic biology, functional genomics, and bioinformatics over the past 20 or so years. And today we are extremely thankful for Sri to take time off of his schedule and talk to us today about this very critical topic. So without further ado, Sri, the mic is yours.
Thank you so much. Thank you for the invitation to speak today. I'm going to be talking about a bunch of work from many people, not only at Octant or UCLA, but amongst a broader community. I'll also say that this is the first time I'm presenting this work, so bear with me as we go through it. I'll also start out with some caveats and warnings. I am not an epidemiologist, a pathologist, a virologist, or know that much about coronavirus biology at all. This kind of came about, as everyone sort of did when the pandemic hit, in what can we do together to help the situation. I think more broadly, we all understand the gravity of the situation, and I will probably make some jokes along the way, just in morbid humor, but I've been incredibly impressed by how the scientific community has come together to some extent without too much leadership, but from a self-organization standpoint. So it's been really exciting and an honor to be a part of that community. So as I mentioned, I'll be talking about a technique we developed called Swabseq that has taken on a direction and of its own, sort of outside of our control, and I'll try to give a broad overview of where things are going. But in general, all of this is due to this virus that you see here, the SARS-CoV-2 virus responsible for COVID-19. And our efforts here have been focused on turning genomic technologies that we've developed for completely different purposes, and I'll talk a little bit about that today, for a kind of a scalable and cheap detection platform for the viral genome that lies within this capsid. I'll also mention some conflicts of interest. I am a co-founder of a company called Octant that might or might not benefit financially from Swabseq if successful, although we're not currently running any tests at Octant. I'm also an associate professor at UCLA, and getting students back to campus is probably useful for all of us, especially at UCLA, which has a huge population of students. And finally, I'm also a parent of 2 young children, and for all of the parents around, our renewed appreciation for elementary school teachers has become clear, and I think we all want to just get back to normal. So with that, I'm going to give some basic points around clinical testing. I think many of you already know the situation in the United States and broadly across the world. I'd say I wrote these points a couple of months ago, and I think they still hold true, although I think the first one's a little off now. But basically, most of the testing we hear about is in the clinical setting, at hospitals, at community clinics, at doctors' offices. in these kinds of settings, it's about triage of symptomatic people. At least 2 months ago, we were probably at the needed scale or beyond the needed scale. But the general thought was, as the economy reopened, that would become problematic, and that seems to be the case now. Today at our company at Octane, we've been trying to get testing from any vendor possible, and right now the turn times are north of 2 weeks, which is essentially useless for the kind of mass screening efforts for return to work. We talk about clinical testing separately from these mass screening efforts for people to return to work, to return to school. I think there's arguments as to whether that's needed or not, but whatever side of the argument you're on, I think a lot of people want to be tested, and there's not enough testing to deal with the demand. Third, there's been a broader push on the test, trace, isolate framework to control COVID-19 through public health measures, but we just simply don't have the testing capacity to do anything like that in the United States. And then finally, this workplace and student testing to bring people back to work or class. Some of this is definitely like, can we stop outbreaks? But there's also just a general trust level of, if I come back to work, can I have reasonable expectation for safety at that workplace? And that's a reasonable thing to want and to have. So the long and the short of it is that we just don't have enough testing. It's not even close. And so how do we think about improving that over time? Today, the dominant test is a quantitative RT-PCR test that detects viral genomes, which are RNA genomes for SARS-CoV-2, and that is usually done through a nasopharyngeal swab or an NP swab that's inserted pretty deeply into the nostril. You collect the specimen, you extract RNA, you do what's called a reverse transcription quantitative PCR that tracks the timing of the PCR to get an approximate copy numbers of the number of viral genomes in your sample, and you return test results. In theory, this should be about a few hours. If you got the sample and had like a complete beeline to the whole testing workflow, but this is dependent upon a lot of things, including just like how do you transport the sample, but also importantly capacity. The reason why samples take more than 2 weeks to return right now is because we're just completely at capacity at either the purification or RT-qPCR stages of this test. So just to dive into a little bit of why this is and why it's the gold standard, and more broadly why limitations are there right now. qPCR's huge advantage is that it's incredibly sensitive. The limited detection in general is around 1,000 copies per milliliter of swab media. So usually the NP swab is taken into 1 to 3 mils of media, usually something called the viral transport media, or sometimes just saline. And that swab that media is then purified and then put into a qPCR test. The limitations today is around one, logistics. So there's just a general logistical problem of how do you accession so many samples at scale. Purification is a big one. So you have to take the media today, for the most part, and purify it using standard molecular biology reagent kits, often provided by folks like QIAGEN and others. The issue with them is that at scale, those become very difficult. Either they require a lot of automation and automation time, or they require people to actually purify those samples. And so that's a huge limitation of this process. There's a general limitation on QRT-PCR machines. These are not just normal PCR machines, but PCR machines with pretty sensitive optics that track the amplification over time. And so these things can be scaled, and many have, I think most notably by folks like the Broad Institute, who've done just an amazing job of scaling these kind of qPCR assays to thousands to almost 10,000 a day. But in general, the throughput per machine is pretty low, and again, it requires quite a bit of automation. One of the big things you've been probably hearing around is the supply chains of everything in this process, from swabs to purification kits to test kits to quantitative PCR machines to tips to automation equipment. All of these things have been put under tremendous stress, especially in the United States. The price right now is pretty high, somewhere between $100, which is a Medicare reimbursement rate for high throughput tests, and $150. The actual test in terms of acquiring it is somewhere between $25 and $75. And finally, the turnaround time is severely limited, but assay time is actually a very small portion of that. As I mentioned before, it's mostly due to this inability to scale and deal with surges. I'm going to talk a little bit about the origin of Swabseq and where it comes from. The technology really comes out of something we've been building initially at UCLA and then at a company called Octant that we started in 2017. I've been on leave at Octant in the Bay Area since 2019, and I'm still living in Berkeley, and the company is located in Emeryville. The company has nothing to do with diagnostics. It's a drug discovery company that's basically the long and the short of it, and I'm not going to expand a ton on this, but we work on ways to generate very large data sets between small molecules, drug targets, cellular pathways, and phenotype that allows us to do things like interpret large-scale genomic data sets or large-scale chemical data sets to better understand drug action and build better drugs. To give you some idea of the types of things we do, we aren't a huge team. We're about 24 people now at Octant, and that's about doubled since a year ago. But we have about 13 scientists, but we produce data sets that punch above our weight. And I'll give you some taste of that. But the thing that basically Swabseq was born out of was a chemical biology platform that's one part of Octant's bigger platform that does the following, which is we engineer large libraries of human cell lines. Sometimes that's 50 to 100 cell lines. Sometimes it can be many more in the case of experiments like deep scanning mutageneses. But the basic idea is we engineer a human cell line, which expresses a particular receptor or druggable receptor, and the pathway by which that receptor signals. And we add, and if it gets activated, it turns on the expression of a reporter. Normally that reporter would be luciferase in a luciferase assay that normal drug screens go through. We add a barcode to that message. That barcode basically uniquely identifies that cell line. So the amount of RNA produced gives you an idea of how much that receptor has been activated, and the pathway that receptor activates, and then we can measure that by next-gen sequencing. And what this allows you to do is do hundreds of thousands to millions of these barcoded lines all in a single dish. One of the big things that we do started from some work started by Eric Jones and Rishi Jaju in my lab, but also Nathan Lubach and Josh Bloom, who are part of this, who are mostly now a part of Octant, where we build libraries of cell lines. We put them into a well of a 96-well plate. In this particular case, this was a set of 50 mouse olfactory cell lines that we had engineered to produce these barcodes. They express a mouse olfactory receptor, and then when activated, turns on some of these barcodes, and we can measure them in high throughput in 96-well plates. And so we spent a bunch of time optimizing this in my academic lab, but then over the course of the last 3 years, really turned this into an actual platform at scale within Octant. To give you an idea of the scales we're at today, Zach individually by himself runs this whole platform. He's a recent graduate of the University of Utah. So the basic workflow is we do an in-lysate RNA amplicon sequencing of a barcoded RNA library of cell lines. Usually we have about 100 or so, or up to 100 engineered cell lines per well. We do this in 8 to 16 3D 4-well plates at a time. The RNA processing portion of this protocol takes about a day. It's optimized to be extremely low touch and inexpensive. We don't have robotics here. This is all kind of done by 96 and 3D 4-well plate, like hand-operated pipetters. And then there's an automated analysis and statistical pipeline on the back end that like crunches all the numbers and puts the results into something usable by scientists. That kind of data looks like this. So this is just, I'm just zooming in on one of these graphs. This is a drug dose response curve. On the left, or in black on those lines, is a DMSO well or a set of DMSO wells. This is one particular receptor, and then adding drug at different concentrations. The top number on the left is essentially an adjusted p-value or a Q-score, a negative log 10 of the Q-score. And so this pipeline lets us do basically very large scale experiments that relate small molecules to druggable receptors at scales that are pretty much unprecedented. But the cool part about this is that you can do this with very small numbers of people and not a ton of automation, if any. So let's see, in early March, we, like everyone in the Bay Area, shut down. And over the course of the next several weeks, we were thinking about, kind of stuck at home and thinking about what to do. This group here, along with many others within Octane, is like, what can we do? Can we just like turn this RNA sequencing platform into something useful? A lot of our thoughts at the time were like, people are like LabQuest and others are probably going to scale, and this is going to be a waste of time. But we were all sitting at home. We thought we might just try it. A small group of us, especially Eric early, and Molly and Aaron kind of got into the lab. Josh, Nate, and Scott helped computationally. And we just asked a simple question of like, instead of detecting barcode in this lysate, can we detect RNA genomes of the coronavirus in unpurified sample? And the general thought was that if we could do that, then we could scale this platform and actually just use the Octane platform or the ChemBio part of the Octane platform as a way to basically do large-scale testing of patient samples. You know, I think one of the things that happened during this time, we started a Slack community. I'll talk about that in a little bit. But just everyone was incredibly helpful, and we were trying to figure out, everyone was trying to learn coronavirus biology and how to do diagnostic testing all at the same time. The picture on the left was pretty fun. In Emeryville, there's a bunch of synthetic biology companies. One of them is Bolt Threads. We were doing a bunch of qPCR optimizations to test different primer concentrations and pairs of primers to see which ones would work. It's kind of some of that data is on the right. And you know, Dan just went one night in the middle of the night and gave us their PCR machine, so a qPCR machine, so we could have more testing or more optimization capacity. So during this time, it was like a pretty intense few weeks, and then things started looking good, and so we decided to open it up and just put it online. The basic idea here is that you can take swabs directly into TE or lysis buffer, rather than going into VTM, and so that initially didn't work. I'll talk about that a little later. We kind of do our process of adding barcodes and in vitro spike-in controls again that I'll tell you about in a little bit. We pull all of that, we sequence it, and then we push the data. In general, as I mentioned, it's pretty simple. We run this process basically week in, week out for our cell line work. We probably run that platform once a week, pretty much every week, 1 to probably 6,000 samples at a time within Octane. So 16 384 well plates is like kind of our max on a normal week. It's fairly cheap. Consumable costs are pretty trivial here. It's mostly around the RT-PCR mixture and tips. It's pretty scalable. Without automation, you could pretty easily imagine getting to 10K per day. With automation, you can get much higher, with most of the limitation being thermocycler capacity. To give you some idea of scale here, a MySeq can get you around 1,500 tests, something like a NextSeq around 5 to 10,000, and a NovaSeq standard flow cell would get you closer to 100,000 tests. again, that all depends on getting the logistics of getting that many samples on board. It's very sensitive. I'll talk about some of the data in a little bit. And in general, at the end of this, which happened in probably early April, we published all the methods and data online and under the Open COVID license, so it's free to use, and also all the computational frameworks, Docker images, the ability to run the whole thing yourself, all under an open source license. I think it's the MIT license, but it might be Apache. In general, the main thing why this is different than anything or fancy, why this is different than almost any other sequencing pipeline has to do mostly with this in vitro RNA spike-in standard. In general, the thing that allows us to not do purification, that allows us to do this in a very low-touch manner, is this standard. And basically, I'm showing you the amplicon that we use in most of our tests these days, which is the S2 gene from the virus. This is the amplicon sequence that we sequence. We only sequence 26 base pairs of that amplicon. And we make an in vitro standard. So this is an in vitro transcribed standard. You can also use it you can also use an RNA oligo, where we basically make the same exact sequence, and the same exact overhangs, except we flip 6 bases at the front end of this. And the reason why we flip them is so we can tell them apart. And we put a known amount of spike-in into each well. And that does a bunch of really important things all at the same time. The first is that it turns a count-based assay into a ratiometric readout. This is kind of well known in the RNA-seq community. You can get this ratiometric readout helps deal with a lot of the issues with like noise around count data. But importantly, one of the big things it does is anything in, say, the NP swab or any kind of unpurified sample, the things that inhibit PCR or RT affect both the spike-in and amplicon in the same way. And so that ratiometric readout still remains constant despite that inhibition. The second thing it does is that it turns like a kinetic assay that real-time PCR is to an endpoint PCR, which is much easier to do. So you can just run this for 50 cycles or 60 cycles or 40 cycles. And at some point, you run out of primer and nothing happens to your sample. And so the nice part about that is that it ends up having a normalization effect. So what determines the amount of DNA coming out of that PCR is the amount of primer that you put into the mix in the first place, because you're going to endpoint. This allows you to call true negatives very easily. So one of the challenges with sequencing-based assays is one particular well that might have a ton of reads will prevent you from having reads in all the other wells. In this case, because everything is going to endpoint, that's not the case, and you still keep the quantitative nature of it because of the ratiometric readout of the spike-in. And then finally, what we find often is that this leads to fewer artifacts. If you have, as many people know, if you just take a set of primers and cycle them for 40 or 50 cycles, you're going to get artifacts. But if you have something in there that's amplifying, it tends to suppress those artifacts. So in general, this is incredibly useful. We use these standards sometimes for the human control, for other amplicons, and this is what allows us to do everything incredibly low touch at Octant, and in turn, what makes Swabseq so straightforward. It's extremely quantitative, so this is kind of some of the data we pushed out in April. This included notebooks to just recreate these figures in a Docker image that was put together by Nate and Scott and Josh. What you're looking at, and you're going to see a lot of graphs like this, so I'll walk you through it for the first time. On the y-axis is the RNA to spike-in control ratio. So this is ranging from 1e to the -4. At 1, it's about a few hundred copies. As you see in the viral RNA copies on the x-axis is a log scale here. What you see is that basically at a few hundred copies, that ratio is 1. And as you go higher, the ratio gets higher. If you go lower, the ratio gets lower. At some point, you start hitting kind of a Poisson noise, as you see at 0.6 copies per microliter, or sorry, per reaction in this case. And then finally on the left is our 0. You'll notice a couple of things, and I'll come back to these things because what we spent the next 3 or 4 months doing is basically trying to suppress the reads that you sometimes see in the zero. So one of the things that you see in a sequencing-based assay is that often you'll get kind of stray reads of the viral genome, even though you didn't put any viral genome in this contrived sample. In this particular case, this is in lysate, not in NP swab, but we have, I'll show you more data over time. So this is kind of how this started. We kind of put in a simple classifier here, but this is what we pushed in April. I mentioned this already. The cost of consumables is pretty low. We basically use this on a NextSeq internally, but we've done this on a MySeq, and other partners have done this on a NovaSeq now. The cost of consumables and sequencing is about $1 to $3, but again, that's not the total cost of the test, which can include accessioning, the actual tubes that this goes into, and quite a bit of other things, including equipment depreciation. As I mentioned, we publish this fairly openly. So we push this out under the Open COVID license. We publish all our protocols, our software, our lab notebooks, our designs, as well as primers and controls. We ship primers and in vitro standards to about, I think at this point, 30 groups out there, and try to build somewhat of a community that where people could just compare notes, and that's been incredibly useful. The team, obviously not the whole company is working on this, and we have kind of our drug discovery goals that are still ongoing, but today, Eric, Molly, Nate, and Josh spend a lot of their extra time on top of their company responsibilities to help support a bunch of groups that have really taken over since then. I'll be talking a lot today about the UCLA group, of which we're also part of because I'm still a professor there, but really led by a larger group that I'll just talk about in a second. The other groups that have really been helpful from the very beginning have been UW, especially Jay Skiering and Jay Shanduri and Leah Starida's lab, and in general, help from the UW SCAN consortium, who've just been incredible throughout this whole pandemic. I think they were the first to find the first community transmission in the United States through the flu study, and that turned into the SCAN consortium to do kind of surveillance testing broadly, and it's become a model, I think, nationwide. Caltech, Sina and Looger at Caltech really helped a bunch with the computational models and rewrote a bunch of the software on the back end, and they have a preprint on that. I'll mention that in a second. There have been corporations, including Helix and Ginkgo Bioworks, that have been providing resources and in general trying to boot up and help the broader community on getting Swabseq working and providing advice. There's been a lot of others, and I'm probably going to miss people along the way, but it's really heartening to be a part of a community of people that have been so open in helping one another get past each of the roadblocks that come about in learning how to do testing. So I'll mention just a couple other things. One is a Slack community that we created. I just checked this morning, it's 931 members today. Lots of details on like methods, EUAs, data sets, protocols, sample collection, lots of news, what grants to apply for, emergency requests, supply chain issues, almost anything kind of practical in nature for those thinking about testing. If you're interested in joining this Slack, please just shoot me an email or DM me on Twitter. There are also many private channels, including that's where we coordinate all our Swabseq efforts, and we have weekly Swabseq calls every Wednesday at 1:30. Again, if you're interested in booting up, that's a really good place to get started. And Molly Gasparino on Octan's side runs those meetings. So I'll mention two things before I get to the UCLA data. There's been a bunch of groups that have helped everyone, including the UW group. This is some of their data from a couple of months ago that Jace produced, where they've been really experimenting with dry swab protocols. So rather than going into VTM, can you send out swabs, return them dry, to deal with supply chain issues and then go directly into things like TE. They've shown really nice quantitation with qPCR. There's a preprint using extraction-free qPCR to do that, but they've also shown that they can do this with Swabseq, and that's helped a bunch of our efforts at UCLA. These are actual CQs of qPCR by extracted RNA compared to direct qPCR swabs on swabs, dipped directly into TE. Again, the y-axis here is the ratio of reads to in vitro standard. The x-axis is the number of spiked in viral molecules, and then on the right are actual CQ values of patient samples, and the coloring gives you the ratio of the Swabseq readouts in, I think, 12-plex. As I mentioned, Lior and Sina, along with folks at Octant, put together a preprint on using the computational backend or developing computational backends that can deal with a lot of different types of sequencing-based assays. And Sina has done just an amazing job of pulling that together, but also helping port it to a bunch of different assays, including Swabseq. Finally, I'll mention a bunch about UCLA. So this is, you'll probably recognize some of these faces. So myself and Josh are actually, Josh is an assistant professor, research professor at UCLA, working with Leonid Krugliak. Leonid is an HHMI professor. We started at UCLA at the same time, and he sort of adopted me into human genetics, which is the department he chairs. I'm not an official member there, but I spent a lot of time learning from the group list here, which is like Jonathan Flint and Eleazar Eskin, who runs computational medicine, but also a bunch of new assistant professors. Yi Yin, who came from Jason Dury's lab, that's in human genetics. Val Arbolita, who's in pathology, and many others. There's like just a big group here that came together and just decided to get started and pouring money into buying the equipment and the supplies and the supply chains, and just pushing through a ton of barriers early on, and even today, to get this off the ground, at least the work I'm describing. And so this really started in March, around the same time when we were kind of getting our first data set showing this was happening. And I will mention Josh. Josh works at UCLA, but also helps us at Octant quite a bit on our back end. So he knew of our process and platform, and so it became pretty, not straightforward, but like very clear that, at least in Josh's mind, that this would work, and in Leonid's and others. And so the idea was just to try this and scale it. And so this initial phase, or called phase one, was about really just optimizing and getting the protocol working. So replicating the Octant setup at UCLA and getting things to work, and I'll discuss that. And then really after we got good at that, starting towards the end of May, moving that into a clinical phase, and I'll talk about what we've been doing over the last month at the end. Let's see. Okay, so the big thing here was like, can we get, so there's the test, which is, once everything is nicely in 96 well plates, can you actually get the test working? And then there's the other big part, is how do you get a bunch of samples into 96 well plates, and how do you accession that at a scale of UCLA? To give an idea of UCLA, UCLA is the largest college or university in California. It has about 50,000 students, about 100,000 people on campus. There's 20,000 healthcare workers. It's the largest hospital system in Southern California, and has over 2.5 million, I think, on order patients within the UCLA health system. So it's a big institution within Southern California. And then if you imagine all the things that COVID affects, universities, health systems, healthcare, and just large, kind of highly dense populations, UCLA is basically an epicenter of how COVID can affect economies broadly. And so the need is very broad, but it's also really unique in that the med school and the main campus, of which I'm a part of in the chemistry and biochemistry department, are completely integrated. And so it becomes very easy to cross kind of the boundary between the college and the health system. So when we started, we thought, let's try a bunch of things. We didn't know what was going to work. Can we try, UTM or saline instead of VTM, the viral transport media? Can we do dry swab? Can we get rid of swabs and go to saliva? These were all things that we started on. There is a massive amount of PCR inhibition for people that know or do PCR within actual human samples. The issues around this were just to increase the cycles or dilute the samples, and we've done both, and we did a little of this over time, just a bunch of optimizations around this. The dilution worked well for saline. In the end, saline was working pretty well, and a lot of our clinical lab specimens moved to saline because of issues around supply chains of viral transport media. We could dilute directly into water and then measure signal to noise of the amplification. What you're looking at on the left is basically different amounts of nasal swabs diluted, either 1 to 2, 1 to 5, or 1 to 10, and you can start seeing this relationship between copy number spiked in and the ratio of reads, as kind of like the last graph. We started doing this. What we found was, like, normal saline definitely has a lot of PCR inhibition, and you can get rid of it over time, but it still remains quantitative if you get the dilutions right. One of the biggest issues that we found, we do see that the synthetic spike-in actually normalizes for well effects pretty nicely. But the other thing that we found a lot of is in the zero well that I mentioned before, there are still a bunch of viral reads, and where are they coming from, and how do we reduce that? Because in the end, that's what limited our limited detection, was the amount of reads pertaining, like, or aligning to virus that we don't know where they're coming from. One of the big things we learned very quickly is how easy it is to contaminate your samples. I think clinical pathology is hard for a reason. It's one of these things where if you're not used to it, you just don't realize how easy it is when you open a tube, for instance, with a lot of virus in it, how easy it is to contaminate other parts of the tube. One way to avoid this is to use PCR mixes that include uracil in them. So there's a small amount of uracil in the DNTPs during the PCR that incorporates within the amplicons. And so your amplified regions then have these uracils, and you can use an enzyme called UDG to basically degrade them. And so if you look at most clinical qPCR methodologies, they tend to include this enzyme. So TACPath, for instance, already includes this enzyme. And by switching to it, we were able to basically vastly reduce the amount of reads of this S2 gene straight off the bat. So what you're seeing on the left is an enzyme RT-PCR mix from NEB called Luna that does not include the UDG, and one on the right, which is this TACPath, which does. And you can see the number of reads drops fairly dramatically on this S2 gene, about 100-fold, or maybe 10 to 100-fold. There were a number of other things too. What we ended up doing, and what the team ended up doing especially, is around, like, systematically finding the areas where noise kind of arises in these sequencing-based assays that included daily bleach and UV treatments of empty tubes, plates, pipettes, diluents, working in a PCR hood for high concentration dilutions, adding a vacuum to the decapper, separate room between pre and post PCR. One of the things we found is the fluidics within the sequencing instruments actually carry over contaminations between runs. Just to show you, adding a bleach wash ended up reducing that again about 10-fold. On the left are just reads of S2 of running just completely empty. We didn't have any virus in these plates, in these 96-fold plates, and on the left and right, this is with a pre-bleach wash, and this is without one on the left. And so the reads again correspond to RPP30, which is the human control, and S2 and the S2 spike in. So anyways, we after systematically doing that, what we found was that the limits of detections are pretty amazing in the assay. This is on the left, purified RNA. The second one is in saliva, so unpurified saliva, and then limited detection on mid-terminate samples from early experiments where we're just spiking in small numbers of copies into each well. So you start getting quantitation that becomes basically as close to as good as you can get in these types of assays. We found here that they're pretty much equivalent to qPCR. We found later during the EUA process, the FDA EUA process, that it's actually quite a bit better in at least purified samples, and we'll talk about that in a second. So that was really the end of June. And July, or towards the end of June and into July, was really putting together the FDA EUA package, the emergency use authorization that would allow testing. That was submitted on July 2nd. The July 6th was the official start of a couple of different, and the 11th was the start of a couple of different projects around testing. One of the strategic decisions we made then was to go after saliva as a sample. And so starting the UCLA Emergency Department saliva study under IRB, as well as the kind of identifying the processes for reliable and extraction-free protocols. And then finally, last week, or I can't tell anymore, but starting July 13th, a big ASH center study, which is a student health center, and actually collecting nasal swabs and/or saliva from students that are still on campus. So what we found was also for clinical samples and clinical specimens that either purified extracts or nasopharyngeal and normal saline, what we found was that the positive patients definitely had high concordance and was easy to separate in our assays from negative patients as quantified by the clinical microbiology lab at UCLA, which runs north of 1,000 samples a day today. We did the NIH limited detection study, or sorry, the FDA limited detection studies. We submitted at UCLA for the Swabseq protocol in purified samples a limited detection of about 250 molecules per milliliter, whereas the typical qPCR is somewhere around 1,000 to 2,000. There have been other FDA EUAs filed with Swabseq that have gone even lower than this 250, down to 125. On the right is just the clinical concordance on 30 patients with negative and positive SARS-CoV-2 status. So I think the last part of this is really around how do we think about extraction-free protocols and filing amendments to this EUA. These were largely contrived samples of mid-terminate nasal swabs into TE, and that's always pretty much looked good. This is extraction-free directly from actual clinical samples on the right. These are for nasal swabs. For saliva, what we find is some of the protocols that have been published openly in medRxiv around saliva using high temperature and TBE methodologies can give you pretty good results. Here we get LODs that are a little worse. We lose some limited detection because we're not purifying. Purification gives you a concentration that we just don't have the luxury of when using unpurified samples. But even then, the limited detection is somewhere pretty close to qPCR of purified samples in saliva. And we just started getting data back. I should mention that was just at the beginning, or July 6th, starting to collect emergency department saliva specimens in comparison to NP swabs, and some of the early data is on the right for that status. And there is concordant. I mean, obviously they're different sample types, so there won't be 100% concordance, but overall we're pretty excited by the results that have been coming in. So the last part of this, and maybe just the last couple of minutes, I'll talk about the test, as we mentioned, is one part of a larger part of testing, which includes clinical accessioning and return of results. That is pretty hard to imagine at scales that are required to bring the economy broadly back to work. The way we think about this is just we're pushing as much of the processing to the end user. So we use directly these matrix tubes, which are compatible with SBS plates and automated decappers, and are also have 2 barcodes sold by Thermo Fisher. They're called matrix tubes. So they're CLIA compliant, and HIPAA compliant. We have a sample collection app. We use, there's been offers from companies like Benchling to help develop the barcode readers to work into our system, and the test results sent to Qualtrics by this barcode. We've been setting up the system over time. You know, we've also been piloting like simpler systems that where end users can just take a picture of the barcodes on their app and then get the return result through the app. This is work by Eliezer-Eskin's group, but also hopefully over time get into the return results directly through ASH. The ASH Center collection has begun, so this is just collecting hundreds of samples from student volunteers, either through nasal or saliva. There are a lot of issues to work out as this goes on, like the barcodes don't always scan, and people have questions about how to actually close the tube or what the volumes are. But in general, the collection's been taking about 5 to 6 minutes per person. And the hope is over time to do this at multiple kiosks across campus. I want to end with this video just to give you an idea of what this looks like. In the lab, we set up a new space that's near complete right now. I'll just start here. So that's just taking the 9612 plate out of the refrigerator. This is just reading the barcodes directly. This is from Thermo, directly in Matrix. To bar reader, there's an automated decapper that just gets put into this machine and takes the caps off. And you have all your samples. We use not automated, but semi-automated 96 well plate pipetters. We also have 384 well plate equivalents of this. Kind of what you're seeing here. The 96 well plate pipetters into 384 well plates, put that into a PCR machine, run the RT-PCR reaction, and then directly throw those whole thing into a sequencing reaction by pooling it. The analysis software is pretty quick after that. I'll end there. There are obviously still a lot of things to work out. That was Lila in the video in Val's lab, and Chaitan and in Leonid's lab, along with Val and Josh, and a few others that have been just really pouring a ton of work into this. This is a large amount of work for a pretty small group of folks. Chongyan Lo is a new assistant professor, Yi Yin, Jonathan Flint, Eliezer-Eskin, Omai Garner at the UCLA Path Lab has been useful in getting clinical specimens. But broadly, just a lot of support and community just pushing, especially the leadership here around Leonid and Eliezer and Jonathan, clearing the way for folks like us to just try to do the science and see if we can scale it. All of that said, I think I'll end there. We still have a long way to go, and I will say that there's other groups that within the community and Swabseq that are pushing through FDA EUA and trying to get this commercially relevant. Yeah, we're excited to support them. The other types of things that folks are thinking about is adding new viruses, so flu and RSV, as well as a few other ideas around making the tests more informative around which strain of viruses that people are being infected by. I'll end there. I want to thank the UCLA team, but also broadly the Octant team, who's spent a bunch of time doing their main work, but also helping support a bunch of these other groups.
Awesome. Thank you, Sri. That was epic. I've got a million questions here. First up, though, Andy has a question, and before Andy asks his question, I want to embarrass him a little bit because Andy invented the ERCC RNA spike ends at Invitrogen. So yeah, he's definitely got a lot of clout. So Andy, what's your question? Oh, Andy, can you turn on your, can you guys, can you spotlight Andy, please? All right.
There we go. There we go. I'm good. Sorry. Yeah, technically not true. I was just part of the team that took the NIST design and put it in a tube. So, but yes, I do know a thing or two about standards and spikes. Great talk. Thank you for putting that together. The video that showed the process in the lab was super helpful. Data, of course, looks great. Is Octant a CLIA lab, or is this thing run through a CLIA lab at UCLA? I mean, you guys are doing the test on site. Who does the paperwork, the billing, the diagnosis, you know, healthcare costs, recovery, et cetera?
Yeah, so Octant is not doing testing. As of this, we are a drug discovery firm that's just trying to support other people doing stuff. Amongst the different teams, there's several that have CLIA labs. The UCLA team is operating out of the hospital right now. The CLIA question is yet to be determined. There's a UCLA, like pathology has a CLIA license that's currently in operation. One option is to maybe extend that over time, but other options include just applying for certification, and they're working through that. Right now, everything is being operated under IRB at UCLA. Sorry, you had a second part there. Oh yeah, the other teams, UW is famously was shut down by the FDA, but then is operating under IRB, but was operating for 2 years. Before that, I think they're operating under CLIA from Debbie Nickerson's UW Pathology. The Ginkgo and Helix, for example, are CLIA labs. So yeah, so I think it depends on each individual partner.
And is there any cost recovery associated with that?
Again, it depends on each individual partner. I think some people at UCLA, it's mostly Leonid and Eleazar just plowing money from their department chair funds, and others just at Octant, we're just paying for primers and helping just ship them. But that's mostly it right now. Obviously, the commercial partners are doing things. I can't speak for them on how they're cost recovering.
Okay, next up we have Connie with a question. So Connie, can you please raise your hand first? Okay, and then if you guys can make sure you unmute yourself. Oh, and Andy, can you lower your hand, please? All right, Connie.
Hi, Shri. I have a question, and that is, do you think this is feasible at this point for a university that's just thinking about starting to test their student body? And is it ready for that?
Yeah, I mean, obviously we at UCLA are trying to make that a reality. It's really hard. Like, obviously I do think that, yes, it's totally doable. I think technologically it's all there, right? But there is a logistical challenge of like who's setting up the kiosk, like who's doing the self-collect, is that going to be approved, who's going to push that through. I think from the back-end technical side, that's all fairly straightforward, other than I think you saw the video. I mean, it's really just adding the sample to a pre-populated set of plates that have like the RT-qPCR buffer and the primers built in, so, and then putting in a PCR machine. That said, the thing that's been challenging that we had to learn from scratch is if you're not used to dealing with molecular contamination, it's challenging and it takes work to like really get that right. So I think you have to put in work to do that. We're hoping it to make it more turnkey and replicable, but you know, I think what we're trying to do first is to get an end-to-end return to college
Yes.
somewhere. And UCLA, I think, is doing a great job of it so far, but it's still a long way to go.
Awesome. Thank you.
All right, next up we've got Kim. And while she's raising her hand and they're turning on her audio, Kim is part of a group here at Janelia working on a LAMP assay for COVID. So yeah, she's got a lot of relevant expertise and good questions. So Kim, you're up.
Hi, that was really great. Have you guys tried going from other collection media types like the BTM or the UTM? And also, do you have kind of a favorite sample type that feels like it's most reliable and behaves the best across patients?
I don't think we have enough data to say like reliably well. I think the broader point is like we're optimizing on scale right now at UCLA, and that has its own limitations. So either it's going to be like there's no way that a small team at UCLA is going to tackle 10,000 like tubes with VTM in them and then turning those into like 96 well plates, right? So all of that has to be pushed to the end user. We thought about ways to do that with swabs, and we thought about ways to do that with saliva. Saliva seems the most straightforward. So we're trying to push forward with that, and that seems pretty reliable. The biggest challenge is there's a lot of RNases in the media, and that's actually not that bad for actual live virus, but it's worse for like the in vitro standards and determining the limit of detection. And so there's been challenges there that I think people have been dealing with, and luckily like groups like the Yale group and the Saliva Direct studies and a few other studies that have been posted on medRxiv have shown ways to get around all of those things and handle those well. So I'd say saliva looks exciting for this kind of mass screening approach. I think the broader point is like, is saliva as good as NP? And our answer to that is like, who cares, right? Like this is not the clinical accessioning and triage of people in the hospital. This is trying to test tens of thousands of people constantly. And if they have barely detectable virus in their saliva, we're probably less worried about them as super spreaders and things like that. So, and I think there's been a series of epidemiological studies kind of showing that's really about test frequency and cost over sensitivity. So that's kind of the at least philosophically the way we've been operating. To answer your question directly, saline has been better than VTM, but that might be dependent upon enzyme choice. So we haven't done a ton of optimization there. There's at least some evidence that like TAC path in particular, which is what we've been using, is very sensitive to VTM over some of the other RT-qPCRs. So we haven't optimized that at UCLA. Maybe some others have within the community, but that's the way we've been thinking.
Yeah, and I think, it's very important to think about ease of collection. If you're going to be collecting multiple times a week, which of course saliva would be easier, even if you're thinking about elementary school kids. But some people complain about the saliva that, some is really thick, some is not as thick. It depends on how hydrated the person is. And I just wonder if that variability, the visual variability, is translating into detection variability.
We've seen it. We definitely started to see some of that variability. I think the 95-degree step with DTT has been helpful in that variability. So like, I think some of that is handled there. I will say that it's something like in the emergency department for very symptomatic people, like it's actually it's been hard to extract any saliva from those people. And so on order half of the people or more tend to just not be able to produce saliva. So I do think that there are problems there that we need to figure out. But for the asymptomatic testing, most people they're all fairly young people that are somewhat healthy, right? So that's been easier. But I think we're still at the early stages of that. I think I showed you the timeline. Like the collection just started about a week and a half ago.
Great.
Yeah, Kim, before you go,
Sure.
didn't you guys find that 95 you got way more RNA degradation than you did?
Yes.
Like,
Yeah. So yeah, so it was better to do, in our hands anyway, to do like a 60 or 65 than to do 95 for longer.
Yeah, I mean, I think it depends on your detection methodology. We actually tried both.
Sure.
We've tried a few of them. The 95C has been super helpful. And like, I think the fragmentation that we were surprised by the 30-minute 95C step, which seems like horrible, but kind of surprised at how well it ended up working, I think.
Yeah, and I think that you're exactly right that the detection, how you're going to detect it, you need to optimize every assay for itself.
Yeah, and I know that like there are some LAMP-seq approaches out there that have, like, some people have data on, and obviously the LAMP color genomics assay has been really fantastic for getting turnaround times and adding more scale too. So that's exciting as well.
In your testing strategy for schools, what's the frequency with which you are hoping to test students? Because it would be interesting to see if with this less sensitive test, how many, for those people who eventually come up positive, how many tests did you need before you detected it?
So I don't think it's less sensitive per se. I mean, I think there's arguments there. B, it's hard with the asymptomatic because, like, to get an EUA on asymptomatic testing is actually pretty problematic right now, because like you would have to get 30 positive samples, right, in an asymptomatic population that's going to be really small. That said, the longer we wait, the easier that's getting, and so maybe that'll change. I don't think we have enough data to answer your question, actually, which is, yeah, I don't think we have enough data yet. I think you saw, if you look closely at that emergency department data, there were some kind of low CT value NP swab, sorry, high CT value NP swab ED patients that tested positive that we called negative in the saliva. But I don't think we're confident enough in our process right now to say that, like, that's not an error, but that could be an answer to some of that, that we might not catch everyone.
In the Yale's data, they did have some that were positive by saliva and maybe negative by nasal.
Yeah, so it was the opposite too, right?
Right. So. But eventually they became positive. There wasn't any sample who never was positive by one of them, right? So it just might be a delay of a day or two or whatever in one assay or one collection type versus another.
Yeah, and I think that's a little bit of the difference between like the back to work and back to school stuff versus clinical, right? Like in the clinical side, like it's important to get it, like as many pre-symptomatic people as possible. And that's the thing that you were trying to get in the back to work, is the pre-symptomatic people that are probably transmitting but have high viral loads.
Exactly. And you want the infectious.
Yeah, and so like, look, if we can't get and I don't like, yeah, exactly as you point out in the Yale data, saliva caught stuff that MP didn't catch. So like, I don't know. I think we're arguing. I think a lot of this, I'm my frustration here, and there's been a lot of good things, but some of the frustration's been around, like, we're arguing around the edges, and we're in the middle of a pandemic, and the entire economy is shut down. Like,
exactly.
Like the things that, so like, I just get frustrated in talking about the edges there sometimes.
Well, it's different if you're trying to diagnose and find out what is this person sick with versus we need to do surveillance and pull these people out, and that's the confusion.
Yeah, and from the standpoint of purified samples in a clinical environment, actually Swabseq is better, in my opinion, or actually from the data, than qPCR by quite a bit. And you could add kind of the differential diagnosis around flu and RSV and other kind of viruses that'll become probably way more important in the fall.
And are you guys, can you get actual sequence data from your end product, or are you doing that?
So So Octant is not involved in any of that. Leonid, Krugliak, Josh, Yi Yin, and others have kind of developed an Arctic-like protocol where they can look at positive samples in LA and along the UCLA.His. So people are doing that for kind of trying to understand where the virus is coming from, and similar to a lot of other groups that have done stuff like that. And I think if for that kind of stuff, I think that the Illumina, who's been super helpful throughout this whole process, has their own kind of full genome sequencing version of using sequencing, and that also gives you kind of more full genome coverage there.
Thanks.
Awesome. So next up, we've got Tom with a question. So Kim and Connie, if you can lower your hands. And I'm going to squeeze in a question while Tom's getting on. If accessioning and other, onerous paperwork related stuff is the bottleneck, but you guys can sequence tens of thousands of samples, isn't environmental sampling like the killer app for this tech? Like, do every place you can find sewage and get a map? There's no paperwork associated with that.
Yeah, I mean, I think yes, but I think the assumption right now in the United States should be that it's pretty much everywhere, and right. So maybe if we are lucky enough to get to the place where we're looking for reemergence, but I think that is exciting. Yeah, I mean, I think more broadly, Swabseq is just, there's nothing specific about COVID. You could do it with DNA or RNA. It's really about reducing the overhead of automation around purification and normalization and things like that. So I think there's a lot of uses for a low cost nucleic acid detection mechanism at scale. Octant is not directly interested in those things, but if other people are, obviously there's a lot of different ways to think about that. And there's really great groups doing that, like Chris Mason's group, Eric Alm's group at MIT, Chris Mason in New York, others are doing things like that. And I think it's been really interesting, actually, surprisingly interesting that the sewage stuff is where I was kind of shocked when I started hearing about that stuff, that you can start detecting stuff before you see it even in the
Yeah, no, I mean, my impression is that it has pointed out a couple useful places to then dig in, that it's not just a curiosity. But yeah,
no, I think I was pretty skeptical at first, but seeing the data more recently, I've been pretty impressed. So yeah, I mean, if there's ways that people want to do it, obviously happy to help any group really wanting to set it up.
Yeah. Okay, Tom's, can you guys turn on Tom? And Tom, unmute yourself.
Hi, Sri. So I heard your comment earlier in your presentation about needing to add, air extraction during the decapping process. And since that's something we're going to be doing also in a fully automated way, I'd really like to know a little bit more about that. Either if you know it or can point me at the people who have been doing that.
Yeah. Well, Tom, you could just come to the next Swabseq meeting like you have been, but no, it's a good question. This is actually something that's come up in the last week as we've been doing the ED patients and kind of at the new facility. We are seeing within 96 well plate contaminations that are likely due to the decapper, and our decapper from Thermo comes with a vacuum line that hasn't been in process. Our new facility doesn't have vacuum in it yet, so we're trying to find a portable vacuum that can pull there. So, I mean, it's part of the machine already has that thing. We just never hooked it up, and it was a problem. So now we have to fix it and try to solve that problem. I don't know how. Yeah, I know, Tom, that you guys are probably doing externally threaded. The thermo tubes are internally threaded. There's just a bunch of this kind of stuff that I don't know if it's going to affect you guys. But I will say everything that we every time we've like done anything different, like we've had to like track down contamination. And so I think any new facility that gets started, just keeping track of it. One of the cool parts about Swabseq, you can do like thousands of tests. Like one of the things we do now is just have an empty plate, because like the differential cost there is like very small, and having an empty plate can help monitor that. So I think anyone doing this, just running lots of extra tests. I do think there's probably a difference with qPCR too, because with qPCR you never open the plate post contamination, or post amplification. But like real patient samples are super blazing hot sometimes, and they can cause contamination pretty easily.
Okay, thanks a lot. I appreciate it.
All right, awesome. Next we've got Sina. Can you raise your hand and unmute yourself? Okay.
Yeah. Hey, Sri, that was an awesome presentation. Thanks for giving it. I was just wondering what your thoughts are on what role the US government should play, if any, in helping to deploy Swabseq.
Yeah, so we've applied for the RADx, I think the day it came out, and did not get it. That was amongst UCLA, Octon, and a few others, including, I think, Ginkgo and Helix are somewhat part of that, and, the Hudson Alpha. So we haven't actually had much government support. You know, I think many here would probably comment on the failure of various groups, including the US government, and like really anticipating scale. There's also just like really simple things like, you know, like the RPP30 thing that the CDC uses and everyone uses, which is that standard human control, is actually a DNA control, not an RNA control. And like, I think the UW group and others are trying to fix that. So it's not actually a good metric for sample quality. I don't know, it's not like, it's not been helpful. It's not been helpful on the asymptomatic stuff. It hasn't been helpful on like different types of like saliva versus, there's just been a lot of confusion. But like, I think it's just a hard environment. People are trying not to make stupid mistakes, and there's not a lot of central leadership. So everyone's trying to find their way. So, I mean, we can throw stones, but so far, I will say, I think there are, and like, again, I don't want to like spill beans for other people, but there are people that are using Swabseq that have gotten big government RADx grants and others for Scaleup on the commercial side. So that's been good, and hopefully that will come out in the news over time.
Cool. Thanks.
Awesome. And Sri, I've got one more. I can't seem to let this issue go of like, if you know, I hate like unused bandwidth in a pipeline. So do you think you could use your massive bandwidth and just toss in like 100 primer pairs for pretty much every pathogen, and just, you know, someone comes back COVID negative, but it's like, you got this other thing, you know? So.
Yeah, multiplex PCR is its own beast. So like there's always like a question of like, are primers going to work together? And there's not like a really rational way to think about that. Like the fact that Arctic works, it's 100 different primer pairs. So it's possible. Sequencing costs and primer costs are a very, very small part of the costs here. So like it wouldn't add anything to the test. We're just trying to like I think it's just, yeah, exactly what you're saying, which is bandwidth. And but I do think like an upper respiratory symptomatic test with RSV and flu would be super useful, right, for differential diagnosis. And like, I think more broadly, there's probably other things, but again, it depends on the sample type. Like, is it upper respiratory? Is it something else? Like you could do it with bacteria for all you know, like in 16S RNA if you wanted to. Yes, you can do all of those things. It's just, yeah.
Yeah. Well, Sri, that was epic. So thank you so much for taking the time.
Thank you, Sri.
Yeah, no, thanks for the invitation, and really thank you for all the talks that you guys put online. I've been watching a bunch of them, and they've been educational for me because this is all new for a lot of us.
yeah, please keep coming back. You know, we're going to have a lot more cool people on.
Awesome. Thank you.
Yep. All right. Stay safe.